So myself, Dr. Amanpreet Kaur, I am a formulation scientist by training and pharmaceutical educator by passion who strive for excellence in pharmaceutical education and research. And today I'm here to present my presentation on strategies for sustainable approaches in drug discovery and formulation development at Chitkara University Faculty Development Program. So let's dive into our topic. First, I'll start with sustainable development. So sustainable development, according to the Brundtland Commission of United Nations, it's a development that meets the needs of present world without compromising the ability of future generations to meet their needs. So the survival of our societies and our planet depends on sustainable world. So this is bit of a juggling act where we are trying to keep three balls always in the air. These include economic growth, social inclusion, and environmental protection. So these are the three main pillars for having a better sustainable future. So sustainable future is possible if we act now and work towards the goals. So these are the 17 Sustainable Development Goals given by UN. You all must be aware. First is no poverty, then zero hunger, good health and well-being. We are always striving to work towards this goal as a formulation scientists and pharmaceutical sciences researchers. Next, quality education. Since we are university employees, we are always striving towards excellence in this goal. Gender equality, clean water and sanitation. Next is affordable and clean energy, decent work and economic growth, industry innovation and infrastructure, reduced inequalities, sustainable cities and communities, which is the need for sustainable living, responsible consumption and production. Next is climate action, which is like, this is very crucial. We have to save our environment. Climate is creating a havoc everywhere, as we have seen with rising global temperatures. Then life below water, life on the land, peace, justice, and strong institution. And lastly, we have to partnership for these goals. So these are the 17 Sustainable Development Goals, and the target is 2030, where we are trying to achieve these goals, and we are very much lagging behind because of the war, conflict, and the COVID-19 pandemic and the post-pandemic effects that world is facing today. So now moving on to the capacities for sustainable development. Sustainable development helps to solve environmental, economic, and social issues in order to create healthier, safer, and productive environment and improve the quality of life. For achieving these goals, we need 6 interconnected capacities. These capacities include capacity to govern cooperatively. Then we need a capacity to measure the progress while we are working towards the goal. Capacity to promote equity, capacity to adapt to the shocks, then capacity to transform development pathways, capacity to link knowledge with action. So these are the 6 very important capacities that we need to build in order to have a sustainable future. So now diving into our topic, that is the drug discovery process. As you all must be aware that the drug discovery process is a long time. It starts with target identification, where molecules, they are selected for binding to a particular target. And this is followed by hit identification. Molecules with desired activity are then taken from hit to lead selection, where the most promising molecules called as leads, they are optimized in the lead optimization stage. Then sometimes there are slight modifications in the drug candidates at this stage. And from here, the lead molecule, it enters into the preclinical research, which is the research on animals. And then 1 in 25 drug candidates is only able to make into the clinical trials. So this process takes a lot of time. And this drug discovery is the initial phase of the drug development timeline, which previously used to take 20 years, but now also it is taking from 12 to 15 years. First 5 to 6 years are in preclinical development, in vitro and vivo testings. Then we move on to the phase 1 trials on volunteers, then phase 2 trials, phase 3 trials, multicentric trials. This whole process takes 12 years. Then there will be drug review, FDA approval, and post-marketing monitoring. And in post-marketing monitoring also, sometimes there are some adverse effects and drugs are being withdrawn from the market. So this whole process involves a lot of energy, infrastructure, time, and resources. So this is high time for us to make drug discovery process more sustainable so that we can work towards a brighter future. So these are the 10 principles of sustainability that are applicable in drug discovery process. First is ecological environmental impact, medical needs, green chemistry, AI and big data, root cause of illness, responsible research and innovation, lean discovery process, cost effective, personalized medicine or precision medicine with the help of biomarkers and bioinformatics. And last is the risk and decision-taking models. So I'm going to take these 10 principles into details in the coming slides. First is ecological environmental impact. We also call it benign by design approach. In ecological environmental impact, we have seen the impact of APIs and their metabolites and the decomposition products on environment. Like, for example, vanishing vultures in India and across the world. Why this has happened? Because diclofenac residues in cattle carcasses have been shown to kill vultures. Because drugs, they are mostly excreted unchanged in the urine. When diclofenac was used for veterinary purpose, the vultures who were feeding on the animal carcasses, they have been killed. Also, another example is exposure of naproxen to light breaks this drug into degrants that are more toxic to the aquatic life than the parent drug. So once excreted, these drugs and metabolites, they pass freely into the wastewater treatment facilities. And there are no designed methods to remove pharmaceutical waste. So that means this is the biggest concern. So what drug discovery scientists or industries have to do, that they have to consider involving the cost of ecological environmental effects into the drug discovery cost, which the proposed cost that they are taking. So like current medicine costs, they do not include this. They are only including the development, discovery, and commercialization phase and clinical use. So there is a need that current medicine costs should include the processes for environmental burden. For this, as per the European Commission report, EU strategy for pharmaceuticals in environment, they have made environmental aspects as an integral part of medical training and professional development programs. This should be part of the training. The scientists, through professional development programs or through programs like this FDP, they need to be trained about the ecological environmental aspects, how to solve the problem of environmental impact. Next is preservation of global biodiversity is very crucial. We have to preserve our biodiversity, which include flora and fauna. They are going extinct. So use innovative tissue culture technologies to preserve the original plants from extinction. Tissue culture is one of the examples to preserve the plants. Next is medical needs. Our medical needs should focus on unmet clinical needs, life-threatening, incurable, or therapy-resistant or neglected diseases. The research should focus on to those areas where there are no treatments available. There are still estimated 6,500 diseases with no regulatory approved therapies. So repurposing opportunities derived from real-world data or AI exploration may catalyze solutions for these unmet clinical needs and accelerate a worldwide socially just distribution of healthcare. So in this part, medical needs, what I'm trying to say is that the medical needs should focus more on the areas which are still unmet, rather than investing on the diseases in which we are already having some therapies. So next is green chemistry approach. Green chemistry, according to Environmental Protection Agency of US, green chemistry is the design of chemical products and processes that reduce or eliminate the generation of hazardous substances. So green chemistry, it prevents pollution at molecular level. It applies innovative scientific solutions to the real world environmental problems. It results in source reduction because it prevents the generation of pollution. It reduces the negative impacts of chemical products and processes on human health and environment. It lessens and eliminates the hazards from existing products and processes. Design the chemical products to reduce their intrinsic hazard. So all the processes should focus on green chemistry approaches. Green chemistry address the ecological impact through waste reduction, resource conservation, and some innovative techniques like bio-inspired synthesis. So there are 12 principles of green chemistry. These are prevent waste, maximize atom economy, less hazardous chemical synthesis, safer chemicals and products, safe solvents and reaction conditions, increase energy efficiency, use of renewable feedstocks, avoid chemical derivatives, use of catalysts, design chemicals and products to degrade after use, analyze in real time to prevent pollution, and minimize the potential for accidents. So this is carbon footprint of pharmaceutical industry. It is significantly worse than automobile industry, as you can see from this graph, that 48.6 is the emission from pharmaceuticals as compared to automobiles, which has shown the emission intensity of 31.4. Similarly, in terms of global emissions also, pharmaceuticals are at a higher rate than automobile industry. So this is high time for pharmaceutical manufacturing and pharmaceutical industry to think about reducing the global emissions and using green chemistry approaches in their processes and production. So with increasing awareness on environmental impact and state of the planet, strategies to make the practice of synthetic chemistry more sustainable needs to explore the following things, like synthetic chemistry should stimulate greener synthetic chemistry parameters have been introduced to evaluate the impact and efficiency through chemical heat and production of waste such as atom economy. And then reduce or the use of solvents by including flow chemistry and synthetic chemistry in water, so that the synthesis is a bio-inspired type of synthesis. So atom economy and bio-inspired synthesis are two approaches that pharmaceutical industry should take into the synthetic chemistry approaches. So ionic liquids, they are green solvents which can be used as drug delivery carriers. This is one chapter I have published in Punjab University, and there some scientists were working in our lab on ionic liquids as drug delivery carriers also. Ionic liquids, they are salts which have a melting point below 100. So these are liquids at room temperature. They are generally made of ions and short-lived ion pairs, unlike the ordinary liquids. They make a unique architectural platform on which properties of cations and anions can be independently tailored, enabling tunability in the design of new functional materials while retaining the core desired features of an ionic liquid. So the beauty of ionic liquids is that they can be tailored. They have tunable properties. So they have n number of applications in pharmaceutical fields. They can be used as solvents. They can be used as drug reservoirs. They are used as drug carriers in microemulsions and in emulsions. They are used as they are used as antimicrobial agents. They can form ionogels. They are used as anti-biofilm agents and anti-neoplastic or anti-tumor agents. Pharmaceutically active ionic liquids are also there. Sometimes ionic liquid and API salts are formed, which shows biological activity. So this was the chapter I have written way back in around 2014 and 15 about ionic liquids. So general salts are solid form, but ionic liquids, they are liquid at room temperature. And I have one case study also to show in our lab. There was development of ionic liquid based microemulsion formulation for dermal delivery of 5-fluorouracil. It's an anti-cancer drug. Here we have used ionic liquid for preparation of ionic liquid in oil microemulsion. So these were the phase diagrams where the formulation was optimized and the particle size obtained was around 10 to 11 nanometers. And then these formulation was subjected to in vitro and in vivo characterizations, and DMBA TPA model was used to study the effect on skin tumors. And very good results were obtained with ionic liquid based microemulsion when it was compared with the conventional formulation available in the market. And another beauty of ionic liquids is that they are very good solubilizers. Most of the drugs, they are not soluble in water. They will definitely go into ionic liquids. So ionic liquids are beautiful carriers. They are very effective solubilizers, and they can also form API salts with drugs also. So next approach is artificial intelligence and big data. Artificial intelligence and significance data automation in drug discovery accelerate the speed and efficiency of the drug discovery process. Synthesis planning, as well as prediction of physicochemical, pharmacodynamic, and pharmacokinetic properties and therapeutic outcomes. So PK/PD comparisons and predictions can be done using AI. This reduces the number of test animals and human or environmental burden. It improves the reproducibility and decrease the consumption of materials. Compound activity data, in vitro and in vivo data, and clinical data, they are growing at an unprecedented rate, affording invaluable big data to deep learning strategies for activity and bioavailability prediction. So these tools are helping to understand the drug repurposing and drug discovery process in a better way. All the data is compiled into big data, then deep learning strategies are applied to get the predictions related to activity and bioavailability. So big data approaches these days are a very important drug discovery toolbox for future. So this is one intelligent drug discovery report by Deloitte, which highlights 5 major uses of AI in drug discovery process. These are 40% of AI is used in molecular library screening. 28% AI is being used in target identification. 17% is used in drug repurposing, 8% in de novo drug design, and 7% in preclinical studies. So these are the 5 major areas in drug discovery and development timeline where AI can be used. These are some of the companies which are using AI in drug discovery process: Pfizer, Takeda, Sanofi, and the list is so long. This data has been taken from ltechsoft.com, which is an AI related company. So these are some real life examples I have selected here to show the use of AI in drug discovery into those 5 areas which we have discussed in the pie chart in previous slide. So for target identification, AstraZeneca is collaborating with Benevolent AI, which is a UK-based AI company. And AstraZeneca has added 2 targets, chronic kidney disease and idiopathic pulmonary fibrosis, to its portfolio for further development and validation. So they are using AI tools and working towards these 2 targets. Then screening molecular libraries. Exscientia and Evotec, they have co-invested first AI-based anti-cancer drug candidate, which is now allowed for clinical trials. The use of AI has shortened the drug discovery period from 5-6 years to just 8 months. Next is de novo drug design. In silico medicine, it managed to create a new drug-like molecule from scratch in just 21 days and another 25 days to validate the compound which is needed to fight fibrosis. They have not given the name of the drug. So drug repurposing, again, Benevolent AI detected existing medicines through AI that could address COVID-19. So It took 48 hours to identify the best candidate, baricitinib, which is arthritis drug. This drug was searched through AI for use during the COVID-19, and finally it was approved by FDA for treatment of hospitalized adults with coronavirus. So this is an arthritis drug, but it was working in hospitalized patients of COVID-19. Then preclinical studies. Curis AI has discovered a drug which is used for Fragile X syndrome, which is most common genetic condition leading to intellectual disabilities with no cure. Here also the name of the drug is not disclosed.
Sure.
So these are some real life examples. Next is root cause of illness. So now in root cause of illness, here the focus of drug discovery should be to treat the ultimate disease, to find the cure for the illness, like developing disease modifying or disease preventive drugs, not merely working on the symptoms. A large proportion of today's drug arsenal elevates symptoms only, rather than working on the underlying cause. For example, mucolytic agents in treatment of chronic bronchitis. So this is high time for the drug discovery scientists to only work on looking for the actual cure of drug. Conventional healthcare system is rather reactionary. It waits until the patient gets sick and then deal with the problem, which is basically diagnosis focused and still largely directed towards symptom management. So the future therapies should maximally target the root cause of the disease rather than the secondary effects. So for example, some newer technologies like PROTACs, mRNA therapeutics, siRNA, CRISPR-Cas9, they are expected to revolutionize the therapies in coming decades. Protein degraders like PROTACs are nothing but proteolysis targeting chimeras. They target the protein degradation and they are showing a very good hope as therapeutic modalities in cancer. In cancer treatment, PROTACs are going to be the future therapies. Next is CRISPR-Cas9. CRISPR-Cas9 is a genome editing tool. These are clustered, regularly interspaced palindromic repeats. Then Cas9 is the enzyme involved in genome editing. This technology has potential to edit the genetic mutations at will. It can cure the diseases, and recently cystic fibrosis, there are some reports that genome editing has a solution for cystic fibrosis where they can remove the gene which is causing cystic fibrosis. So there are some ethical concerns also with CRISPR-Cas9 technology. Then is our mRNA therapeutics. These are the new class of biologics that can be used to encode any protein of interest in vivo for prophylactic and therapeutic vaccination. And they can also be used to encode therapeutic antibodies. The classical example of mRNA is COVID-19 vaccine by Moderna and Pfizer. And also siRNA is also a new approach which are used as gene silencing agents. They are also working towards the actual goal because with siRNA, if it is encoding for a particular gene, that gene will be silenced in post-transcriptional modification. So next approach is risk and decision-taking models. Optimal sustainable decisions taking the complete life cycle into consideration. So we have to think about the whole life cycle of the drug, starting from procurement of raw material to the end user and how it is going to impact the environment and how the waste strategies are there. So we have to shift from cradle to grave model to cradle to cradle model. In cradle to cradle model, the process is in continuous manufacturing type of stage where there is no end. The waste is being utilized for some other process. So this was a concept given by Michael Brogart and William McDough, cradle to cradle approach. The sustainable development is working towards moving from cradle to grave model in product development life cycle to cradle model. With predominant paradigm shift in drug discovery consists of identifying and designing selective ligands for specific target. This can be done using understanding of molecular cellular network, which is done through quantitative system pharmacology approaches. We can improve the translation of preclinical discoveries into clinics. So use of network pharmacology, quantitative systems pharmacology will help understand the drug pathways and the targets in a better way. So new computational approaches such as sustainability metrics are anticipated to guide critical path decisions. So this is the risk and decision-taking models. Next is use of biomarkers and bioinformatics to support precision medicine. Now, what is precision medicine? First, we see what precision medicine is. Precision medicine is giving the right treatment to the right person, taking into consideration his individual disease profile, his general health, genes, effect of genes, environment, and lifestyle. So in the conventional treatment models, one treatment fits to all. Like all the cancer patients, they are given one drug only. But in personalized medicine, the data from the cancer patient is taken, blood sample, urine sample, tissue analysis is done using omics approaches, new diagnostic techniques. His genetic profile is being studied, his lifestyle is being studied, his general well-being is studied, the environment in which he is present. Then the therapy is tailored as per the need of the patient. So personalized medicine is going to be the future. This is going to save cost as well. And then what is the role of biomarkers? So biomarkers and bioinformatics, they support precision medicine because in order to obtain the information on the basis of what the therapy is to be designed, we need bioinformatics and biomarkers. So this will reduce ineffective medicine use, which is burdening the patient, society, and environment when we are giving one drug to all the cancer patients. Drug discovery phase should incorporate genetic and metabolomic markers that can be used to stratify patients according to disease risk and optimal drug therapies. So there should be a space in drug discovery phase that we have to include the genetic and metabolomic biomarkers profile into that. The phase should also consider drug non-adherence. There are some cases where drugs are not adhering. Patients are not adhering to the therapy. There is no response. This leads to inefficient use of medicines. This should also be considered into the drug discovery phase. Therapeutic drug monitoring of drug metabolites and recent advances in adherence enhancing devices, including smart drug delivery and accessory devices, may increase the optimal use of drug. So therapeutic drug monitoring is crucial, and some tailored devices like smart drug delivery or accessory devices should be used in precision medicine. So this is what we have understood about precision medicine. Now, how this biomarker and bioinformatic data is being used? What are these omics technologies in drug discovery? We have to use the multi-omics approach in order to understand the biology of cancer and design the therapies in personalized medicine. So what are these approaches, multi-omics approaches? So if we recall the central dogma of molecular biology, DNA, then transcription leads to RNA, translation leads to protein, and then there will be metabolites in the body. This is not part of the central dogma. Central dogma is still here only. So DNA. In genomics, we will study the genetic profile, the whole genetic profile of all the genes present in the body, identification of variants. This is done through DNA sequencing, including coding and non-coding regions. And there is one more thing in genomics, that is epigenomics. Here, evaluation of non-DNA sequence changes such as methylation or methylation in the DNA. These are epigenetic changes that are taking place due to cancer. These can be identified using genomics approaches, using next-gen sequencing technologies. Next is when it moves to RNA stage after transcription. So that's why this technique is called transcriptomics. Here, evaluation of quantity and quality of RNA. In transcriptomics analysis, everything is done on RNA. It is the analysis of RNA genetic makeup. So next is after translation, proteins are formed. The study of whole proteins, evaluation of protein map network, production and modification. All the studies related to protein comes under proteomics. And then metabolomics. This is the evaluation of endogenous or exogenous metabolites profile in the body. So this is how I want to differentiate between the different omics technologies, and all these approaches are together used to study the genetic makeup of a cancer patient or any diseased patient. So these are collectively called as multi-omics approaches. They have a crucial role in drug discovery and drug development process. So microarray comparative genomic hybridization, this is also one technique which detects the chromosome copy number or changes without the need of cell culturing. So this technique, you don't have to do cell culturing. Using a gene chip, the information can be obtained on the chromosome copy number. So these are diagnostic techniques. Next is using proteomic patterns. Proteomics is fast and powerful discipline aimed at the study of whole proteome or sum of all the proteins of an organism. So this is done using CellDTOF, MS, mass spectroscopy, protein chip, and then diagnosis. This will give you the entire information from a serum sample, and you will get the information on the protein analysis in your diagnostic report. So this was whole of the technologies that are involved in precision medicine, like bioinformatics and biomarkers or molecular diagnostic techniques. Now, what are the challenges and barriers in using this approach? Initial investment cost is very high for these type of sustainable practices. You need to have a lab where you can have apparatus for performing the sequencing studies. You need to have a mass spectrophotometer. You need to have all the advanced technology. So initial investment cost is high. Resistance to change from traditional practices within the industry. Still there is resistance to change. And then limited availability of sustainable resources and technologies. There are regulatory hurdles also and inconsistent policies across the regions. These things are being used in the developed countries like in US and Europe, but in our country we have some regulatory hurdles also. There is no infrastructure is available. Only the high-end labs are having sequencing analysis. So these are some of the barriers. For sustainable development, we have to invest in these technologies also, so that ultimately our drug discovery timelines are reduced and we are in a better place. So next is cost effectiveness is also one of the approach in sustainable drug discovery. The medicines should be cost effective. There should be equal as access of medicines to all. So if we are saying in first goal that there should be no poverty, free access to healthcare, so that means medicines should be cost effective also. Societal acceptable prices, adjusted reimbursement, intellectual property regulations, regulatory data protection, and market exclusivity for repurposing. These are all the things that need to be considered while developing cost-effective drug discovery program. Then initial discovery phase is the phase which ultimately determines the price. So in the initial discovery phase, we have to include all these things like ecological environmental impact and the use of advanced technologies like bioinformatics and biomarkers. So considering all these things into the initial discovery phase and then working with the regulatory agencies and intellectual property regulations and work towards making the therapies cost effective and available for all. Next is lean discovery. Implementing a lean discovery approach in drug discovery involves swiftly and efficiently discovering the right medicine, utilizing optimal experimental designs. So this approach focuses on systematic experimental designs. By following reproducibility principles and recognizing the need for effective regulation in drug discovery, we can ensure its pivotal role in success of drug development and overall life cycle. The right questions need to be answered in a logical order. so that the drug selection process is as efficient as and sustainable as possible. So lean discovery approach is nothing but using design, using a systematic approach, like we are using QBD, DOE. So use a systematic approach so that the process is efficient and sustainable. Then sustainability by responsible research and innovation. Ensuring sustainability in drug discovery requires responsible research and innovation, particularly in addressing the increased difficulty and costs associated with finding and validating novel therapeutic targets. This involves fostering academic sustainability, government support for equitable access, and integrating global sustainability evaluations into the authorization of medicines. Next is, this highlights the need for comprehensive education in drug discovery sustainability within academic institutions. So responsible research and innovation means everyone has to come together. The educational institution where we are getting the training towards the drug discovery, their sustainability should be included. It should be part of the regular curriculum, and government should support, provide access to the data, to the resources, so that responsible research and innovation can be achieved. Next, there are some other ways to understand sustainable drug discovery approaches. This involves signature mapping. Signature mapping involves there is a relative comparison of certain distinct features of drug with the target. The examples of using signature mapping is prednisolone and topiramate. They were identified using drug disease similarity approach, using the correlation between gene expression signature of the drug and that of the disease. Next is pathway matching. Pathway matching, this is done generally using network pharmacology approaches, quantitative pharmacology approaches. Example is pranlukast, an asthma drug, leukotriene receptor 1 antagonist, and amrinone, a phosphodiesterase inhibitor. They are used for congestive heart failure. These drugs are developed using pathway matching approach. Next is in silico screening and molecular docking. Molecular docking studies have identified many drugs. Example here is mebendazole, antiparasitic drug and inhibitor of vascular endothelial growth factor 2. This is also a mediator of angiogenesis. There are so many other examples also of using in silico screening. Then is retrospective analysis of clinical data. Retrospective analysis of clinical data includes, there is large amount of data available in clinics with industries. There are a lot of pharmacovigilance data, adverse drug reporting. All that data needs to be compiled and integrated, and then understand the data to develop the new drugs. For example, terbutaline sulfate, anti-asthmatic drug candidate, was arrived for treatment of amyotrophic lateral sclerosis from retrospective clinical data analysis. Then high throughput screening. This includes the discovery of disulfiram, a drug used against alcohol abuse. Through high throughput screening, it is turned out to be a selective anti-neoplastic agent also, with proven research using genome-wide gene expression studies. So high throughput screening is also used for discovering the new indications or new treatments for the drug. Next is drug repurposing, very important. Drug repurposing is also called drug repositioning, reprofiling, retasking. So this is an effective and practical complementary method for selection of alternative therapies which are either discontinued, abandoned, or on any investigational drug or any new chemical entity, with a parallel study of new metabolic pathways and protein targets. Drug repurposing reduces the environmental impact. It helps in conservation of resources, decreased waste, lower cost, faster time to market, and address the unmet needs. So these are some examples I have put in this table for drug repurposing. The very old example is aspirin. In somewhere in 1800s, it was discovered by Bayer as an analgesic, but later on, and today also, it has been used as an antiplatelet agent. Next is sildenafil, developed by Pfizer. Initially, it was an antihypertensive drug, but later on it was used for erectile dysfunction. Then minoxidil, again developed by Pfizer. It was initially proposed for gastric ulcers and it failed in that indication. Also for arterial hypertension, where it worked. But recently, FDA has approved 1% minoxidil cream for androgenic alopecia or alopecia areata. So its repurposed indication is alopecia. Thalidomide, very old and tragic drug, I can say. This was developed by a German company, Cami. So it was launched as sedative tranquilizer, and in around 1956-57, Fucomelia was one event in the history when it was given to pregnant women. The kids with distorted limbs were born. So then this drug was withdrawn. Then it was repurposed. It has shown a major role reversal in decades, like it's now being used for erythema nodosum, leprosum, and multiple myeloma. This ENL is some rare form of blisters that occur in leprosy that can cause blindness also. Here, thalidomide has got a repurposed indication. Valproic acid, developed by AbbVie. It is a broad spectrum anti-epileptic drug and used for bipolar disorder. Its repurposed indication is anti-neoplastic agent. So these are some of the examples. There are so many examples in literature. Now this is a small case study which we have done in DIPSRU, and it is a work in progress. We are still not through with this. It is an insight into the repurposing of phytoconstituents obtained from Delhi's Aravali Biodiversity Park as antifungal agent. So what we have done here in Delhi, there is a biodiversity park in Vasant Vihar. We visited that park to take the information on the available plants, whichever plants are available in the biodiversity park. We have gathered that information through the help of plant scientists, which are deputed there. Our team visited. We have taken some photographs also and some information. Then we have list out the constituents which are already reported in literature in all those plants. We have prepared the list of constituents. Then our approach is working towards developing antifungal agents, herbal antifungal agent. Plus, we also want to target the problem of antifungal resistance. So we have used a dual target approach. So dual target approach, for that we have selected two targets. One is steryl 14-alpha demethylase, which blocks the conversion of lannatosterol to ergosterol and leads to accumulation of aberrant sterol compounds and depletion of ergosterol in fungal cell membrane. Second, we have taken HSP90, which is a highly conserved protein chaperone involved in circuitry of multiple signaling pathways in response to stress. So both these targets we have selected. Target proteins were HSP90 and steryl 14-alpha demethylase. Then through the information on the plant constituents from Aravali Biodiversity, we have performed the virtual screening studies and selected the hits. Then we have identified 3 or 4 compounds depending upon the docking score. And the next part of the study will be biological activity, in vitro activity, in vivo activity, and then we will be ultimately developing the formulation. Till now, we have published till identification of compounds through docking. So this was the plan. 45 plants were studied. Then 1149 ligands were selected against these two targets. Virtual screening was performed using Schrödinger suite 2016 software. Then we have selected 83 hits against both the targets, and finally 3 constituents were selected: quercetin, isocathamidin, and brevinone B. Then MMGBSA studies and MD simulation studies were performed in order to validate the stability of these ligands. Next step is in vitro and in vivo characterization, and finally we will be developing a formulation. So this docking studies was performed by one of our computational scientists, Dr. Kalicharan Sharma, who was present in DIPSRU at that time. Currently, he is not there with us. He has joined ISF Moga, but we are working as a team in achieving this herbal antifungal drug discovery pursuit. So one more thing I would like to emphasize from this case study is that we need collaboration. We need multidisciplinary approach in drug discovery process. We need a computational chemist. You also need a pharmacologist to understand the network pharmacology. Then in order to develop a formulation, you need a formulation scientist. We required the help of government also, Aravali Biodiversity Park, the plant scientists present there. So research is not always in one hands. You always need to have an interdisciplinary or a multidisciplinary approach. So collaboration is the key for that. I cannot perform docking studies myself, but if I will collaborate with some computational chemists, he will do the best solution for me. So this is the approach where we all have to collaborate. So this was our approaches for drug discovery. Next is formulation development. In formulation development, how we can work towards sustainable development. So in formulations development, what we are doing, we are trying to improvise the already existing drugs which are showing some faulty formulation design. There is no bioavailability, there is unwanted distribution, misdirected delivery. Sometimes they are ineffective and toxic. So all these conventional approaches have these problems. So we are focusing towards development of novel and sustainable drug discovery. So for this, we are using nanotechnology is the high-end approach we are using. So we are developing nano carriers in order to effectively deliver the drug through skin or through oral route or any route. So liposomes, niasomes, transferosomes, SLNs, NLCs, these are all the nano carriers of choice. So we have to think about the excipients that we are using in order to develop these nano carriers. Like lipids are very good natural, biocompatible, and biodegradable excipients for developing these nanocarriers. These days, lipid nanocarriers are in very much demand. Like they have a long journey from liposome, drug-loaded liposome, targeted liposome, then mRNA vaccine, this Moderna and Pfizer vaccine. This is lipid nanoparticle only. Solid lipid nanoparticles, NLCs, cubosomes. So they have a great journey from 1955-56 when liposome was discovered till 2021 mRNA vaccine. So lipid-based nanocarriers, they have a beautiful journey, and lipids are biocompatible and biodegradable. They are better solutions for developing the nanocarriers rather than going in for polymers. For me as a formulation scientist, I always prefer lipids, though there are some biocompatible and biodegradable natural polymers available, but lipids are my favorite when I am thinking about developing any nanocarrier based formulation. So this is one company, Gatfose. They provide lipid excipients for topical use, for oral use, for rectal or vaginal use. They have all the excipients. Plus, this company is also working towards sustainable development. Their overall objective is to reduce the total greenhouse gas emissions by 25% by 2030. So I'm not trying to promote any company here. That is not my purpose. I just want to make everyone aware that their lipids excipients are very good. They are of high quality and they easily provide gift samples. You just have to send an email and give a brief about your research profile, what you are trying to do, and they'll be happy to send you any samples. So this is one company from where you can obtain all those excipients. Next is our formulation development processes. They need to be energy efficient. We have to optimize the manufacturing process for energy efficiency as well. We are optimizing it for our quality attributes, for our process parameters, but one energy efficiency should also be there. You utilize renewable energy sources in production. Work towards minimizing the water consumption during manufacturing. Reduce waste generation. Cut energy consumption through efficient processes. Use eco-friendly solvents and practices, chemicals. Implement waste reduction and recycling programs in laboratory. So these are some of the ways how we can make the manufacturing process efficient. Then sustainable raw materials. Use sustainable and natural raw materials like plant-based ingredients, bio-based ingredients, marine resources, biomimetic synthesis. recycled and upcycled paper, algal and fungal extracts. These are some sources of sustainable raw materials. Then waste reduction strategies. Implement green chemistry principles, optimize reaction conditions, use catalytic processes, employ solvent recycling systems, and use process analytical technologies throughout the monitoring of your process. Use biodegradable materials for packaging and partnership for sustainable packaging. Educate the staff on waste reduction. So waste reduction strategies should work towards disposal, reduce, reuse, recycle, and recover. Next is packaging. Green packaging for pharmaceutical manufacturing. Utilize biodegradable and compostable materials. Design packaging with recyclability and reusability in mind. Reduce packaging waste by optimizing the packaging design. Explore innovative packagings such as plant-based plastics or mushroom-based materials. Incorporate eco-friendly printing techniques and inks to further enhance the sustainability. Now, next, how pharma companies can become more sustainable by implementing continuous manufacturing instead of batch manufacturing. First thing, efficient packaging to reduce the transport space, invest in green chemistry, adopt water conservation techniques and wastewater treatment processes. They should be adopted in-house, shifting towards renewable energy, implementing a sustainable procurement policy, design drugs with end of life in mind to enhance recyclability and reduce environmental impact. Implement take back programs for unused and expired medicines to prevent pollution and misuse. So this is one example of Sky Pharma. This is a French company. It is a contract manufacturing company. They have implemented sustainable development into their corporate policy. So Sky Pharma Board of Directors, they have developed Sky Pharma Sustainability Development Committee through volunteer program. They have sent an email that who wants to participate in such type of policy or such type of committee. Then 15 voluntary and motivated employees from all departments they have picked, and they have made a plan that they are going to spend 1 or 2 hours a month on sustainable development. So 5 points they have included is energy cost reduction, where the plan was painting the production building roof with light color to reduce heat effect, managing lights using timers and LEDs, photovoltaic panel installation. Then for water recycling, they have bio-waste using compost, daily administrative waste using sorting garbage cans, specific waste from our production activity. Preserving sites, flora and fauna, installation of beehives they have put, inventory of flora and fauna species and implementation of actions to preserve them. Then environmental awareness, creating awareness tools for environmental protection, training sessions on environmental impact of our actions to the employees, and sessions to visit the sites, hives. Then they are also working toward ISO 14001 certification project, working on obtaining this certificate. So I am not promoting the Sky Pharma here, just an example that they have implemented sustainable development policy in-house with their board of directors and including all the employees. So this is one way to do at an institutional level. So according to Xtalks reports, 10 most environmentally sustainable pharma companies of 2023 and 24, Pfizer, AstraZeneca, Sanofi, Merck, they are all in the list. Now, what is the social impact of sustainable development? Improved community relations and public perception, job creation and support for local economies through sustainable practices, health and well-being benefits for employees and surrounding communities, contribution to addressing societal challenges such as climate change and resource depletion. This is the overall impact of sustainable development. So this is a glimpse of some of my journey so far. So this was Young Scientist Award I received in Australia at 5th World Congress on Controversies, Debates and Consensus in Bone, Muscle and Joint Disease. This was about a rheumatoid arthritis treatment, developing lipid-based nanocarriers. So next, in US, I got Academic Excellence Award or Biotechnology Student of the Year in biotechnology training that I got there, and graduated with Certificate of Specialization in Biotechnology, because after doing PhD in pharmaceutical sciences, when I went to United States, I have seen a huge gap in my training and skills related to biotechnology. I was not trained in doing genomic studies because we don't have the access to any of such equipments. And the whole focus of pharmaceuticals is shifting towards biopharmaceuticals. So biopharmaceuticals, you need to have the training on biotechnology and molecular biology. So this was the opportunity I got to add those skills into my profile. So this was the St. Louis Community College campus where I got the training and where I worked also. This was my team at Center of Plant and Life Sciences at Bridge Park. So I also got the chance to work in molecular biology-based plant research at a biotech company, CTC Genomic. It was basically ag-tech company, agriculture technology, where we have grown sugarcane in the greenhouse. US is very cold country, and sugarcane is crop in Brazil, South America. Brazil is having high temperature, so it was very difficult to grow sugarcane in greenhouse. We have to maintain the temperature of 40s and 50s inside the greenhouse. So then I also got the opportunity to work on protoplast extraction, because protoplasts are used for doing the transformation experiments in plants. So it was a great learning experience. Now after returning from US, I have got the opportunity to work at Delhi Pharmaceutical Sciences and Research University. This is first pharmacy university and third in the world. Our university has also developed first innovative oral intradialytic product, because we have in-house incubation and innovation foundation. Then we have also developed first herbal medicine which targets ACE2 receptors for treatment of COVID, Coroquel. This is also an interdisciplinary work with the University of Missouri in US and DIPSRU here. So there are so many people involved in this team. Next, we have first Center for Advanced Formulation in our institute. This was the award I received here, Bharat Shiksha Gaurav Puraskar. So we have an inbuilt precision center also through one of the government grant, and we have inaugurated our precision center in 2022. We have NGS technologies here. Some of the projects are going on, like aeogenomics, post-COVID genomic analysis, cancer panel through Biomark Corporation Canada. These are some of the collaborative projects. So these are some of my journey in DIPSRU so far: welcoming the governor, precision center training, panel discussions. So this is again one very interesting, and we have organized last year Chintan Shivar in collaboration with the DSIR, where I got the opportunity to show my lab developed products at Tech Bridge. So this is one anti-aging formulation of hyaluronic acid. This is for arthritis pain. So this is our research team, PhD student, showcasing our product to the industry partners. So lastly, sustainable committees and a green infrastructure plan. We need sustainable communities to develop. For that, first you have to develop a community vision, establish goals, access assets and opportunities, plan for long-term operations and maintenance, develop strategies for local funding, identify federal government resources, then factor brownfields and hazardous waste sites into planning, identify approaches to add green infrastructure, monitor and measure the progress, and organize stakeholders. This is a small chart to understand how we can develop sustainable communities and have a green infrastructure plan in mind. This creates the basis for social and economic development and ensure equal access to all the natural services. So the take-home message from this discussion: first is de novo drug discovery and drug repurposing. They can support each other to make pharmaceutical innovation more sustainable in terms of resources, time, and setbacks. Then capacity building for sustainable development. We need to have capacities in order to develop sustainably towards our goals. Next, use of multidisciplinary approach in higher educational institutions in giving the training, designing the curriculum, redesigning and designing, considering sustainable future in mind. Then green infrastructure helps to keep the environment clean, solve climate problems, natural problems, clean water, soil, air, improve the quality of food and life, and also cradle to cradle approach. Next is use of ionic liquids and green solvents in pharmaceutical processing. They are beautiful solubilizers. Selection of lipids and natural lipids for fabrication of nanocarriers for drug delivery technologies. Use of multi-omics approaches and precision medicine in designing future therapies. We need to use multi-omics approaches. Then use of AI and big data for reducing the time and money time and money in the drug discovery timeline. So that means here is an opportunity for all the faculties like us to understand AI and big data. This is high time for us to invest our resources in understanding the use of AI and big data, because data science, AI, multi-omics, these are the future technologies. We have to continuously add skills to our profile. So these are some of the take-home messages. Thank you so much. And with the UN slogan, it's high time for us to act now towards the sustainable goals